today's episode of the HealthTree podcast for multiple myeloma, a show that connects patients with myeloma researchers. I'm your host, Jenny Ahlstrom. We'd like to thank our episode sponsor, Karyopharm, for their support of this program. Now, before our show this morning, I'd like to let you know that we will be launching what we call HealthTree 2.0 on October 23rd. When we started this, and this podcast is actually exactly 10 years old, I wondered, how can patients help accelerate a myeloma cure? How can we contribute for these amazing researchers, one of which you will hear from today? And over the last 10 years, we've built over 14 software tools to do precisely that. So we have an education pillar of tools that include this podcast and our news website, our HealthTree University, our roundtables. And we have a pillar of, our second pillar of programs is community building, so we connect you to each other. So you can use our HealthTree Connect social media network or find a myeloma coach, or we'll have a growing number of regional communities that you can help volunteer for in the future. And then the third pillar is our HealthTree Cure Hub data portal, and that's really where we want to invite you to help participate and help these researchers accelerate a cure faster, because when we share our myeloma stories and the investigators are able to do surveys and studies and data research using our real-world evidence, we get closer to that cure. So we are super excited to be able to share that with you. Look for that live and virtual event, mostly virtual event, on October 23rd, and please go ahead and register for that. Now, on to our show. Dr. Paul Richardson, you have been so kind to participate in this mid-year review. We're a little past the mid-year review because we had technical challenges last time. But to help us understand the many advances that are being made in myeloma, and we do this type of review twice a year because there's so much happening. So, Dr. Richardson, welcome to the program.
Jennifer, it's an absolute pleasure and always wonderful to connect with you and have the opportunity to share some of the excitement around important recent advances over the last 6 months or so in myeloma, which, as you point out, Jen, they just keep coming, which is so wonderful to see, but also tempered by, the reality that we've got lots of work to do, and whilst progress is made, again, the challenges, as you and I have discussed before, remain very real.
Yeah, very much so. Well, let me give a short introduction for you before we get started, because we have a lot to talk about. And be thinking of questions that you want to ask at the end, and we will open it up for caller questions at the end. So Dr. Paul Richardson is R.J. Corman Professor of Medicine at Harvard Medical School and the attending physician of the Division of Hematologic Oncology and the Multiple Myeloma and Bone Marrow Transplant Service at the Dana Farber Cancer Institute. Dr. Richardson is a clinical program leader and director of clinical research for the Jerome Lipper Multiple Myeloma Center at Dana Farber. He's also the chairman of the Multiple Myeloma Committee for the Alliance and Alliance Foundation for Clinical Trials, and the Myeloma Steering Committee member of the National Cancer Institute. He is editorial board member for publications like Clinical Cancer Research, Journal of Clinical Oncology, Journal of Oncology, and many, many others, and is a distinguished myeloma specialist who is highly influential in the new drug approval process, having led many phase 3 clinical trial efforts to get many of the drugs that you're familiar with, bortezomib, and many immunotherapies, daratumumab, carfilzomib, selinexor. You've heard of these drugs and maybe use them to market. So we're so appreciative for his efforts there. He has received the Ernest Butler Lecture and Prize from the American Society of Hematology for his translational advances and achievements in enabling clinical science in myeloma, and he's also recognized and appreciated by the patient advocacy community as the recipient of the Robert A. Kyle Lifetime Achievement Award by the International Myeloma Foundation for his work that's resulted in significant advances in research, treatment, and the care of myeloma patients around the world. So Dr. Richardson, thanks again, and we will just jump in. Maybe we just want to start in kind of how a patient moves through their myeloma journey, because there's a lot to keep updated. So in the newly diagnosed myeloma setting, what are the changes that you're seeing? Are we going from triplets to quad therapies for newly diagnosed patients?
Well, Jennifer, thank you. That lovely introduction. Just a one little quick, if I may. I finished my 10-year term as the Alliance Chair, and I just want to acknowledge the new chairman, Dr. Sargis Mani. took over in that important role about a year ago or so, and so having and obviously doing great things. And during that time, actually, obviously we did during my 10 years there, we obviously saw the determination trial come through, the CLGB 100-104 lenalidomide maintenance trial, which to many of our listeners, it was actually the platform for the FDA approval of lenalidomide maintenance. So I'm very kind of grateful to my alliance colleagues for that, and in particular Dr. Phil McCarthy, because we ran that trial, started that trial way back in around 2005. And what's so beautiful is that I have patients still on that study, and we're now, where are we, 2023? It's amazing. So it points to the real progress been made. And in addition to determination, which we can touch on in a minute, with the leadership of Pete Voorhees and the Alliance team during, about 9 years ago, we started the Griffin study. And it's so relevant to just what you asked, because the Griffin study, which we were very grateful to be partnered with a fantastic team at Janssen, we were able to do a large randomized phase 2 trial to document clearly that the addition of daratumumab to RVD, which is our workhorse, as you know, really was amazing in terms of results. And so the Griffin study laid the stage for now quadruplets with a combination of RVD plus daratumumab, and for a randomized phase 2 run from an intergroup study, the alliance with AFT support and Janssen partnership, which was absolutely key to be very clear, we were able to generate so much information. So RVD Dara has become, a gold standard, if you will, in the upfront setting, recognizing that phase 3 trials are still cooking, but this large phase 2 provided a breakthrough to insurers and to provide access for our patients to the importance of a quadruplet therapy. So to kind of share for the patients on the call today the sort of rationale, the idea is that obviously if lenalidomide, which is a fabulous drug we've worked over the last 20 plus years on, is our Navy, and bortezomib is our Air Force, and dexamethasone is our Army, daratumumab becomes, the Marines. And the addition of the Army, Navy, and Air Force with the Marines has been a game changer. And of course, we mustn't forget the Coast Guard of zoledronic acid and drugs like it, but nonetheless, sort of quadruplet approach plus the fifth drug of anti-resorptive therapy, upfront treatment for patients has been transformed. And the big question is, how much does transplant add to this? And I think that in the transplant-eligible group has been very important. And one of the most interesting aspects of all of this is the evolving space. And I want to especially acknowledge my colleague, Dr. Hani Hassoun, who presented again from the Alliance platform our results from determination in subgroups at the European Bone Marrow Transplant meeting in Paris earlier this year. And what Hani showed, which was quite remarkable, is that whilst transplant in younger eligible patients clearly results in progression-free survival benefit, we don't see with longer-term follow-up overall survival benefit yet. We may, but not yet. But most importantly, what we see are subgroup benefits, and specific subgroups do particularly well. And most importantly, some groups actually don't benefit from early transplant. So this was really very important data. And what Hani showed was the fact that certain high-risk patients, particularly those who may have 414 as a cytogenetic abnormality, may benefit from early transplant in terms of progression-free survival gain. But interestingly, patients with standard risk and/or, for example, a higher body mass index may get less benefit from early transplant. And what was so interesting, Jen, is that he sort of drilled down deeper on our work regarding ethnicity and race. And what we've identified in our African American patients is that actually for African American patients, for actually reasons of pathobiology, the access to free medicines, everything was equal in determination. So we were able to ensure excellent access, and we had 20% participation, or close to. It was actually 19% participation from our African American patients. And because of that wonderful participation, we were able to ascertain that, in fact, in the African American population, early transplant, particularly for people who have higher BMI, may actually be not associated with the benefit that we would have expected. And it points, I think, to a pathobiologic mechanism that we're hoping to better understand. And my colleague Jeff Sonder and other colleagues from the determination team are hopefully going to be presenting, we hope, breakthrough data on this at the ASH meeting coming up in San Diego in December. So more to follow, but real excitement about, choices for patients, that you don't necessarily have to have one size fits all, and that quadruplets are really making a difference. And I would be remiss in not acknowledging the incredibly good work of investigators such as Ola Lundgren, who've looked at carfilzomib-based strategies combined with lenalidomide, dexamethasone, and daratumumab, KRD-DARA. And Ola has specifically identified the success of KRD-DARA as an upfront approach. And similarly, Luciano Costa has led the master trial where we've combined KRD-DARA with transplant and seen that equally to be very promising. But I think what's so exciting about Ola's data is his very high rates of MRD negativity as a research clue pointing to clinical benefit, without having to go to transplant, which I think for patients is a wonderful choice to potentially consider, recognizing that clearly high-dose therapy remains a standard of care for the younger patient with myeloma, but the point is that you have choices, and I think for patients going forward, that's absolutely key.
Well, absolutely, and I love what you're saying about personalizing care also from the beginning. So because what you're saying is we can subgroup patients, and not everybody has to be treated in the same way. And personalized medicine doesn't necessarily need to be all genetics-based. We can look at other things like race or body weight or things like that and decide. I think that's so fascinating. Can I ask about a further question about just go ahead.
Well, Jen, I was just going to say, before we dive into that, I think you've raised such an important point, because multiple myeloma is truly multiple myeloma, and I think you're so right, Jen. Subgroups really matter, and what we're learning is that these have real, pathobiologic correlates. So one obvious example is age. And as we talk about upfront therapy, I think it's important just to acknowledge incredibly important advances for older patients too, because what we've realized is that, you've got 3-drug platforms like lenalidomide, dexamethasone, and daratumumab, the so-called RD-DARA, that performs extremely well in older patients. But what's also equally important is that we're also looking at the quadruplet of, for example, RVD plus DARA, but with the RVD adjusted for the older patients so that it's better tolerated. And exploring that as an exciting option for patients is really very interesting, because there are a number of trials that are looking to sort of tease this out better. But my point simply is that in the old days you would be thinking, as you say, of one size fits all. If you're transplant eligible, that's the way to go. If you're not, we look at other things. We're getting much more sophisticated in our approach, and I think age is an important part of that, and I simply emphasize it because it's not just frailty or perhaps inability to tolerate treatments that are so intensive, but it also points to, immune exhaustion or absence thereof, and how that may be very important for tailoring treatments to improve outcome.
Yeah,
Thank
absolutely.
you, Jen. I'm sorry.
No, it's such an important point, and this goes to something that I always say in every show almost, is this is why you need a myeloma specialist on your team, because if you are making therapy decisions at the beginning of your treatment or at relapse, and the person treating you does not understand all these different nuances or how things could be adjusted, you are just not going to do as well, and you won't have as high a quality of life. So I always suggest that patients try to consult with a myeloma specialist as they're making treatment decisions, and then get your infusion at home. But that's just such an important point that I want to make. Okay, let's talk about personalizing care a little bit more. You mentioned in the transplant idea about high-risk patients with maybe 414 patients doing better with transplant, but for other higher-risk patients, are there other approaches that you're seeing either in clinical trials or just right off the bat? If you see a high-risk patient, younger or older, what's your go-to strategy in trying to manage that?
I'm so sorry, Jen. So in terms of you're speaking of high risk, you mean, correct?
Yes. Yeah. Are there other approaches beyond that transplant approach that you would look at for high risk patients?
Yes, no, absolutely. Well, I think importantly, the thing about risk is to understand its complexity, Jen. We're realizing, again, from determination, we learned that there's many nuances to what high risk means. And this field is constantly evolving. For example, 1Q amplification or significant gain is now recognized as a higher risk factor. We've also recognized that there are differences between the implications of 17p deletion and actually partner abnormalities with it, as well as, say, for example, translocation 414. So I think there are a lot of nuances to interpreting risk, which I think is a first important point to share with our audience. I think what we've learned from recent data in terms of high risk is that we have in our armamentarium some very exciting drugs. Obviously, we've had the revolution of immune therapy with, of course, CAR T being dramatically impressive in the relapse setting. And we also have bispecifics making a tremendous splash in the relapse space as well. I think what's important to recognize, though, is that there are other small molecules, not in the cellular or T-cell redirection space, which may be very valid in the high-risk setting. And, for example, selinexor, which is an XPO1 inhibitor, an oral agent, is one very good example of that, which targets 17p deletion quite specifically in the way that it works. And the clinical data support that, as well as the laboratory studies that have been done. I think beyond that, we also, of course, have, our currently available agents with different aspects to their activity, but carfilzomib is a very potent high-risk adapted proteasome inhibitor, and so is bortezomib in its own right too, but ixazomib also seems to target 17p in the frailer patient for whom an oral approach may be preferable. And in the same token, in the space of antibodies, it's very clear that the monoclonal antibody isatuximab, for example, is particularly useful at targeting 1q amplification, really breakthrough data from the ACAMA trial led by Dr. Philippe Moreau establishing that. So I think there's a variety of options that we have to take on high risk. And again, Jen, this sort of pivots back to our previous point that we're able to tailor to patient need. And certainly from the point of view of a younger patient who's transplant eligible, clearly in a variety of trials, not just determination but in others, we've shown that higher risk patients do benefit, it appears, from early intensification. I think what's important, though, is to note that much of this data doesn't necessarily reflect the impact of quadruplet therapies and what they may be doing. So we just have to bear that in mind. But having said all of that, we've got good options for our patients, and I think the promise of the cellular therapies in particular being used early, to my mind anyway, is a very attractive and exciting new avenue of research.
Yes, we're definitely going to talk about that in a little bit. I want to ask you a couple more questions before we get to that. So minimal residual disease testing is kind of gaining in popularity to potentially even become a new clinical trial endpoint. How are you using MRD testing to manage treatment after, first set of therapies or your first line therapies and then at relapse?
It's a super question. I think that MRD clearly is such a useful tool in the setting of research. I think, Jen, what we have to be a little bit careful about is how we interpret its use in the clinical space as standard of care. And I think there's definitely a role, in my opinion, for its use in adjudicating decisions around particular approaches. For example, again, just pivoting back to the determination trial, we showed that MRD negativity was associated with better outcome regardless of what you did, whether you had a full induction remission strategy with early transplant or you delayed it. If you were MRD negative, patients appear to do really well regardless. So this is an important principle that MRD may help guide treatment choices. But I want to emphasize other trials led by really outstanding investigators have shown similar responses, similar outcomes as well. I think MRD therefore has great utility in the research space, no question. In the clinical standard of care space, I think the one caution I would have about it is the importance of being sustained. In other words, just one MRD measurement alone, be it positive or negative, needs to be taken in context. And I think the question of whether or not it's a sustained result, be it positive or negative, is relevant to making treatment choices. And above all, the whole picture has to be taken into account. But I think the role of MRD continues to grow. I personally see it as a remarkable step forward and a very important tool in our armamentarium to tailor therapy. So more to follow, but certainly something that may help guide treatment choices. And one thing I would say, Jen, is it's very useful, in my opinion, after induction remission therapy in newly diagnosed patients. Its value later when we're dealing with relapse and a slightly more unstable situation in terms of clonal evolution and so on of the disease. There one has to perhaps use it a little bit more judiciously because, as you and I have talked about before, Jen, you can see MRD negativity after CAR T, for example, that can be very reassuring initially, but then actually unfortunately not be associated with, may not be that stable. It may actually not last very long, whereas in other settings, MRD negativity can be much more sustained. So I think it's a complex equation, but one that is very exciting to have the ability to use in the appropriate setting.
Mm-hmm. Wonderful. And for people who aren't aware, MRD testing is just a more sensitive way of testing the monoclonal protein after treatment. Well, let's talk about also personalizing care when it comes to 11-14 myeloma, because that's the only type of myeloma that has a specific drug. Do you think about using venetoclax early on, or do you kind of have patients with 11-14 go through their first therapy and then use it later, or what's your strategy there?
It's a super question. Jen, I just want to clarify one thing on MRD, just to realize it is also a look at the level of the cell. That's the key, right? It's not just a look at the monoclonal protein, it's a look at the level of the cellular activity of the disease. So we have currently complete response, stringent complete response, and then when we drill down to MRD negativity, it's times 10 to the minus 5 or times 10 to the minus 6, so ability to detect disease at a cellular level. And we either use this highly sophisticated flow cytometric method, which we christen Euroflow, or next-generation sequencing, which is built by Adaptive Technologies. And I think they both have great value, but certainly in the U.S., Adaptive Technologies is a very strong, it has a very strong presence in this area and has proven to be very, very helpful certainly in our clinical trials. But just to clarify that for the audience, because, again, it complements the assessment of protein, but you are quite right, there is another entity which is called heavy chain assay, which actually is a very tighter drill down on protein, and that's a research tool that's developing very quickly, or heavy light it's called for short. In any event, pivoting back to your other question, which is absolutely spot on, about whether or not to use venetoclax in translocation 11-14, I would agree with you very much about the role of venetoclax in this setting. You raised it so nicely before, Jen. It's all about subgroups, and the point is that this particular genetic subgroup of myeloma, 11-14, it means that for patients, it means that in your disease you have the translocation. It's not obviously in you as a person, but it's in your myeloma that you have this translocation of these chromosomes. And what that means is it translates to a particular series of events within the tumor cell that can be interfered with by this remarkable oral agent, venetoclax. And what's so interesting is that in 11-14 disease, particularly in relapse, it's particularly helpful. Unfortunately, it's not yet FDA approved, although I'm hopeful that may change relatively soon, but it is FDA approved for other cancers, so we're able to use it off-label. What's so interesting, Jen, is that if you use venetoclax in a non-11-14 translocated patient, i.e. their disease does not have it, the outcomes can actually be not nearly as good, and in fact deleterious, which is why I think, in fairness to FDA, they've been very careful with the approval here, because obviously we've got to be very clear it belongs to the 11-14 patients. And you asked, I think, a key question, should we be using it earlier? I think certainly in first relapse it's justified. Up front is probably still the realm of protocol-directed treatment at the moment, because obviously upfront studies to date have not been deployed and or read out, although although I do think in early relapse it makes sense to deploy a very straightforward oral agent that works really well in my experience, although one does have to be careful about infections and blood counts when you're administering the therapy.
Okay, wonderful. Well, you mentioned first relapse, and so let's talk about that a little bit. What strategies do you look to for first relapse, and then we'll maybe talk about earlier use of CAR-T.
Yes, well, I think in early relapse, it's a lovely question, Jen. I think in earlier relapse, obviously what's so interesting, and it's an evolving treatment landscape, as the quadruplets have become so effective in the early treatment setting, when we now look to first relapse and second-line therapy, we're having to think in terms of treatments that our patients previously would have embarked upon much later in their natural course of their disease. Now, the good news here is that if you've had upfront treatment, especially with either a triplet or a quadruplet, the likelihood is, with or without transplant, your disease control will be a number of years, hopefully many years. Then when you relapse, the question will be, what do you deploy? And typically what we use are a variety of drugs that may recapitulate some of what you've had in terms of drug class, but importantly be different. So, for example, if bortezomib has been used up front, the use of carfilzomib in first relapse makes sense, and vice versa. If carfilzomib has been used early, the use of a proteasome inhibitor like bortezomib, but rationally combined with something else, which enhances response. So one thing that's clearly happening is that we are using proteasome inhibitors, immunomodulators, and next-generation approaches in early relapse. But what has arrived that's made such a difference in this, first relapse space has been the impact of BCMA targeting therapy. And of course, in that regard, probably the news since our last podcast together, Jen, has been the excitement around CAR-T therapy, as exemplified by CARTITUDE-4, which was presented as a plenary session at ASCO in June, and then published in the New England Journal almost simultaneously. And showed that the use of CAR-T in first relapse for younger, fit patients who were eligible for this approach really resulted in unprecedented disease control compared to standard approaches that were used for the control group. Recognizing for those patients who participated in the control group, they could obviously have access to CAR-T at a later date. So really exciting data, but I think it heralds really a new way of thinking about early relapse, which is how do we integrate BCMA-adapted strategies or targeted strategies in those patients.
And I had the opportunity to sit in that ASCO session, and it was incredible. The data was really so impressive, really unbelievable. So when you look at, I know that these companies, Bristol Myers Squibb and Janssen Oncology, are looking for earlier approval in these 1 to 3 lines based on some of this data. Is that, do you think it's more effective because the patient's T cells are fitter, or what do you think is the reason for, or the disease is just not as sophisticated by that point because you're using it earlier lines? What do you think is the reason for the better impact?
Well, I think all of the above, right? I think that, as you've so nicely framed it, Jen, I think patients are better able to tolerate the platform, which is important, and I think also their immune exhaustion may be that much less, and therefore this may be very powerful. But I think there are some important caveats. I think obviously we know that targeting BCMA with CAR-T therapy is highly effective in the majority of patients. But we also have to be aware that there is a side effect profile, and sometimes, rarely fortunately, but sometimes it can be really quite serious. So I think there's a lot of work to do to continue to sort of show the reproducibility of this approach in earlier disease. But I think you're absolutely right, Jen. This heralds a new era, and I think that as we make CAR-T therapy more available, which I think is another key point, more accessible, and at the same time that much safer, it will, I think, hopefully bear tremendous fruit going forward. But I think we would be mistaken in saying, it's the only thing, because at the end of the day, obviously CAR-T, it isn't, right? I mean, there are many other options for patients.
Right. Patients relapse and...
Well, not just that. I think it's we have to remember the median age of myeloma is 70 in terms of its onset. My practice has many patients in their 80s and early 90s, clearly an intensive approach such as cilta-cel may really be not wise in such an older, frailer population until we know much more, particularly about the central nervous system toxicity, and at the same time, the ability to use other approaches. Because as you and I have said before, Jen, together, treating multiple myeloma is truly a marathon. It is not a sprint. And, what we do early may really impact on what we do late. So I think we need to be careful. But I also think, having said that, you're quite right that this obviously, the other exciting thing in my view in early relapse is going to be bispecifics. I mean, you have the fantastic approvals of elranatamab this summer as a new BCMA-targeted bispecific T-cell engager with this T-cell redirection quality. And what I liked about the elranatamab data was, the subcutaneous approach. It may be that much easier to therefore give as an outpatient, which is critical for accessibility. And at the same time, it's got every 2-week injection times, and these make life for patients so much better. And at the same time, there may be some differences in infection profiles and complication rates, although I think one has to be careful with that. Having said that, the activity is 60 to 70% response rate, which is very much along the lines of what was seen with teclistamab, and which is the other big bispecific that's approved for BCMA. And then, of course, to add on to that, we have the approval of talquetamab, which is another bispecific T-cell engager, but importantly for our audience, it targets GPRC5D. Now, please don't worry about the long name, but that is a different target than BCMA. It isn't associated with the same BCMA-related toxicities, although it does unfortunately cause skin changes and changes to nails and some epithelial mucosal surfaces. But I think what's most important is that it is a new target, and again, the data there look very promising, again, around a 70% response rate. So these are really remarkable agents. But again, I want to stress, I think we've got lots of other ones, right, Jen, we can talk about, and I'm looking to you to guide me there, because obviously we could spend the whole conversation on bispecifics and CAR T, but if you'd like to touch elsewhere, I'm very happy to so do.
Well, I want to focus on just a few follow-up questions, if you don't mind, because it's a very big discussion point. So first, why is BCMA such a great target for multiple myeloma?
Well, because B-cell maturation antigen, as the name sounds, is expressed in myeloma almost universally. But it's important to realize that it's a downstream marker in the ontogeny of myeloma. So we would be remiss if we said that if you target BCMA, you don't have to worry again. Unfortunately, that's not true. BCMA targeting is highly successful, and despite the success of the CARTITUDE-4 patients, we do know from earlier studies such as CARTITUDE-2, where updated data were shown, great results, but at the end of the day, patients still unfortunately are relapsing. And speaking for my own patients who've been through various BCMA CAR Ts, one of my patients very recently, she had a wonderful experience in terms of tolerability with a different platform. It was a CARSgen CAR T, again targeting BCMA, but she enjoyed 3 years of remission with it, but then unfortunately has relapsed. But nonetheless, it's been well worth that 3 years because she's had an excellent quality of life. The important point is, what do we do now? So we have to realize, that these relapses occur. But anyway, pivoting back to your key question, BCMA matters because it's ubiquitous and because it's a marker that we realize, like CD38, we can exploit accordingly. There are other ways of targeting BCMA, not just bispecifics and CAR T, but also antibody drug conjugates. And in particular, the example here is belantamab mafodotin, which provides a very different approach, which we can perhaps talk about in a moment. But I think it's important to note that BCMA is not only expressed by plasma cells, and that's the problem. It is expressed in the central nervous system, and that is very relevant because some of the toxicities that we see, particularly the fortunately very rare but very real entity of late Parkinson's, which unfortunately to date hasn't been manageable. When it occurs, it's proven generally incurable. These are the kinds of things we have to be on the lookout for. But I want to stress, fortunately, these types of serious toxicities are very rare now, especially if we use use the platform earlier. So I think this bodes well. And of course, I would stress these late-onset CNS phenomena are not seen with bispecifics. One of the downsides of the bispecifics in the BCMA space is infection, and that's one of the things we have to be a bit careful about as well.
Mm-hmm. And going back to the neurological issues with some of the BCMA-directed CAR T therapies, is there a way of telling ahead of time, like maybe is it family history, or is it that somebody already has a toxicity issue? Is it because patients have high tumor burden, or what do you think is causing, or do we know enough about that yet or have enough data about that?
Yeah. Yes, it's a super question, Jenna, and I would say to you, definitely we know that higher tumor burden can be an issue, that's for sure. So the lower the tumor burden going into CAR T therapy, the less likelihood of serious side effects. I think also you're right that there may be vulnerabilities related to underlying medical issues that may steer us away from necessarily targeting BCMA in that way, perhaps looking at GPRC5D or FcRH5 as another receptor target that one might pursue. So I do agree with you. There are opportunities to be more tailored in that regard. But I think we're learning a lot, and I think the great news, what I'm so impressed by, is the whole enormous effort being put behind cellular therapy and the remarkable energy and scope of all the studies that are being done. So I think that we'll learn a lot relatively quickly about this as we go forward. But I think, to your point, we do have to be very thoughtful about patient selection. Mm-hmm. Certainly in my own experience, serious toxicities have been usually in my older patients. Having said that, I have seen some surprises in my younger patients, so I think one has to be careful. But I think at the same time, one cannot argue with what you saw at ASCO in June, which was so remarkable from the CARTITUDE-4 team.
Yeah, it would be so amazing to know that ahead of time, and then you could give someone a BCMA bispecific antibody and then follow it up with a different CAR T with a different target. I mean, this goes back to what we talked about at the beginning with more personalized approaches, and that would be incredible if you had that information ahead of time. And probably you don't right now, but we just need more data.
Yes, and I think I can help a little bit here. I think in the field, the data that we've seen more recently suggests going for BCMA first and then going for GPRC5D afterwards is an algorithm that people are following. But you're quite right, Jen. Is that based upon a really strong scientific rationale? Perhaps not quite yet. I think there's some clues, extramedullary disease and so forth, other features which may be guiding those sequences. But I do think that the success of the antibody proteasome inhibitor, immunomodulator, small molecule platforms provides us with an ability to be very kind of strategic. And so I think, going to target BCMA and then weaving in next generation targets makes great sense. But I think what's important for patients to hear is that, as we said a little bit earlier, it's very important to take the long and strategic view. And I think that, there are some patients in whom you have to pull, your big guns early because their disease is so aggressive and so difficult to treat. There are other patients who can enjoy success for years and keep these more sophisticated and complicated approaches in reserve and use them when they need them.
Mm-hmm. And you talked about accessibility, that they're more accessible. Can you speak to that? Because I know that at the academic center facilities, they were getting 1 or 2 slots for some of these CAR Ts, and I think that's changed, right?
Oh, it most certainly has, particularly in the area of clinical research, that for sure, for protocols, and also on the commercial platforms as well. I think that this has all gotten much better. I think having said that, and the bispecifics have helped tremendously because they've taken a lot of pressure off the system so that, we don't have the waiting list that we had to deal with of old, which was so troublesome and so complicated to navigate. I think having said that, the off-the-shelf itself construct is really important to embrace. For the majority of my patients, they do not want to come in the hospital. They do not want to face complicated regimens, and if at all possible, they would much prefer everything outpatient. And I think this is incredibly important when one thinks of access to communities, and particularly vulnerable communities. So I think the strength of some of the recent research as well has been in the availability of not just infusional strategies or hospitalization requiring strategies, but strategies that can be deployed in the outpatient setting. And obviously, I think the bispecifics will increasingly move into that area where we don't have to hospitalize patients for step-up dosing. We'll be able to do this as an outpatient. But equally, I think, and CAR-T may get there too as well, with a strong outpatient component. But I think what's important, Jen, is really to emphasize the value of some of the true outpatient strategies that are, I can see a patient in the clinic one day and start their treatment either that same day or the very next day. And I think those are very important to have in the toolbox as well.
Right, and that's this difference between, so maybe we want to talk a little bit about sequencing and also different strategies. So fixed or continuous duration of these bispecific antibodies, especially with what you're talking about, like the infections being an issue, or other reduction strategies. So do you want to address what's happening on the research side to determine, do we do bispecific antibodies on a continual basis? There were other studies that were doing two bispecifics together, and another one that I think you had mentioned, the ALNUC. I wasn't familiar with that one, so let's dive into bispecifics a little bit more.
Yes, of course, Jen. I think, obviously the continuous administration of these drugs in a relapse refractory patient in which other options have become exhausted makes complete sense to do in case they're
right.
relapse. But earlier, you're absolutely right, the fixed durations do. And I think I appreciate you mentioning it. Beyond, obviously the success of teclistamab, the impact of elranatamab now emerging, and then, of course, we have elnuc, as we call it for short, elnutamab, which is coming shortly on the heels of all of these antibodies, as well as other ones. There's a phenomenal AbbVie platform as well that is showing great promise in clinical trials. So I think there are lots of these agents coming. I think the important point is you're right, there needs to be convenience and fixed duration, potentially adding to the benefits. I think as we think more about BCMA targeting, this issue of infection is very real. Everyone needs intravenous immune globulin, IVIG. Some real-world experiences in Europe, from expanded access programs were particularly sobering and showed some of the challenges when you don't have highly sophisticated prevention strategies for minimizing infection. There was particularly striking data from some groups in Eastern Europe which were really sobering and told us, you've got to be so careful with infections. So I think we need to be aware of how we best integrate that into our treatment paradigm. So yes, you're quite right, Jen. Lots of research looking at this. I suspect we'll be looking at fixed durations because patients would love that anyway, and then perhaps a conversion to oral therapies that can maintain remission in a rational fashion.
Yeah, that's so. It'll be so exciting to see what happens. And I know there are, I don't know, 6 or 7 bispecific antibodies. I know Regeneron has one, AbbVie has one. It's just expanding exponentially, so it's amazing. Do you want to circle back and talk a little bit about belantamab mafodotin and just provide a little update about that? Because I don't know, when we talk about sequencing strategies, if a CAR-T stops working or something like that, can you take a break, do a different type of therapy, and then go back to a BCMA target, maybe like that, or just provide an update on where they're at with their data also?
Well, I think, belantamab mafodotin as an antibody drug conjugate exploits BCMA but then delivers a warhead, which then triggers immunogenic effects within the disease that then enhances the body's immune system to destroy the myeloma. So, and it doesn't carry with it the risks of either cytokine release syndrome, which can be really challenging for patients, particularly with cardiovascular issues and so forth, and it doesn't carry with it the risks of central nervous system injury, which of course can be fortunately rare, but can be really serious when they occur. So I think there's real hope this approach, in my opinion, because again, I think it's a matter of, all hands to the pumps. It's never one versus the other. We'll need all of these agents. The biggest challenge for belantamab was the ocular toxicity, and the really good news there is that we've really figured this out. Dose and schedule really matters. You can dose belantamab mafodotin every 6 to 12 weeks now and see efficacy, particularly in combination, and lower the dose. This also has implications because you can imagine here, Jen, a patient can come to the clinic once every 6 to 12 weeks even for their infusion, obviously be monitored in the interim, but nonetheless be on an oral agent partnered with this in a way that could be highly convenient and also effective. So these are exciting approaches that have been refined as we've tried to better figure out how best to use belantamab. But I think we would be remiss in suggesting that it has the same sort of power in terms of response rates that, say, for example, teclistamab has, with a 60 to 70% response rate. That's not what we see with belantamab as a monotherapy. It's about half that. But when we combine it with other drugs, that's where we see it show its promise. So when you combine belantamab mafodotin with oral agents like pomalidomide, important to remember that the Algonquin study in Canada showed a median progression-free survival to belantamab combined with pomalidomide of 24 months, which is remarkable if you think about it. It's 2 years, right? So this approach clearly has promise. Now, the challenge has been really more of a regulatory one, that the accelerated approval for belantamab mafodotin had to be withdrawn because of the failure of DREAMM-3 to meet its primary endpoint. This study was a head-to-head comparison of belantamab mafodotin versus pomalidomide and dex, and as someone who had the privilege to develop pomalidomide and dex, which I love as a drug, it's a very good platform and it's great combined with other agents. To see PomDex perform better than we expected was great, but obviously, unfortunately, belantamab did not meet its primary endpoint in that particular phase 3. But, Jen, we've been here before. Carfilzomib, in its first trial, randomized trial, after the accelerated approval, actually didn't meet its primary endpoint. Fortunately, Aspire came in shortly afterwards and led to its full approval. And look at carfilzomib, recognizing its cardiac... Right. It nonetheless is a game changer. I mean, it's improved survival. It's a high-risk disease. It's incredibly important. So we've been here before. So I think the next set of generation of studies from for belantamab coming through include DREAMM-8. The DREAM studies are the acronym for the studies that belantamab mafodotin uses as their global effort continues. And I'm hoping that studies like DREAMM-8, and there's another one called DREAMM-7, as those come in, they will help be kind of Aspire-like and bring belantamab back to access and approval to patients so that we've got access for patients to it. Because certainly in our own practice, and we're leading trials of belantamab combined with other novel agents as well as our own expanded access program, we clearly see a role for it. And to your point is, where is it? Well, it can be used, ideally, if you're going to go for BCMA early, go probably in the appropriate patient for either CAR-T first or bispecific in a young, healthy, well, otherwise fit patient. But in a frailer patient for whom CAR-T or bispecific is really problematic for a variety of reasons, the appropriate use of Bela with careful ocular care and monitoring, as well as a rational combination partner. In our experience, we've seen some remarkably good results. So I think, again, it's an example of why we need all of these agents, not just a select few.
I agree. Yeah, I agree. Every myeloma patient might use every drug at some point. And let's talk about others in the pipeline. And I know this time goes by so fast, and we have to talk about so much because this is a review. But do you want to talk about the cell mods like mezigdomide and iberdomide?
Absolutely, I'd be delighted, because again, this boils down to oral agents that are truly off the shelf, and above all, then provide access, and particularly to vulnerable communities, be they rural, frailer, older patients, minority communities who do not want to come into hospital, for example, for logistic and socioeconomic. So this is, to my mind, a priority, and we're so pleased with how the cell mods have performed. Iberdomide is one to touch on first. It's important to remember these are not, as one colleague said to me, are they glorified IMiDs? They're really not. They are in that sort of broad spectrum of drugs that are immune active and immune modulators, in a broad sense. But if you look at them at a molecular level, they're bigger molecules than the classic thalidomide, lenalidomide, and pomalidomide. They're actually bigger in their size, and they engage the whole of the cereblon E3 ligase complex. So they literally lock in a way that the immunomodulatory drugs classically don't. And they've been specifically designed to do this, which I think is an enormous acknowledgement of the skill of the medicinal chemists at originally Celgene who put this together. It's now obviously under the umbrella of Celgene BMS. And essentially, mezigdomide and iberdomide engage the cerebral E3 ligase complex, do so powerfully, and trigger what I think is really the key of their activity. They are powerful degraders. They degrade Aiolos and Icaros, which are two key transcription proteins that are fundamental switchboards in B-cell pathobiology. And they not only kill myeloma cells, but they also activate the immune system. And, in our team, we've christened mezigdomide CAR-T in a pill, because you can give it, and it's so powerful that it actually can dramatic responses in both the tumor and at the same time in the cellular components of the immune system. And by doing that, you get, just in an oral therapy, really encouraging results. So it was a privilege to be part of the mezigdomide phase 1/2 team, and we published our data in New England Journal, actually in hard copy just a week ago. So the timing of this podcast actually, Jennifer, in some ways is perfect. And we got a nice editorial with it from the New England Journal and written by a super scientist in Australia. And Dr. Short really did nail it. I mean, he nailed it. There's some beautiful diagrams for anyone who can have access to it. I'm happy to send it to you if you like to have them to share.
Yeah, I would love to share it. I would love to.
I'll happily send those to you, Jen. Just because Dr. Short's editorial is really very thoughtful. I think the diagrams are lovely. The important point is that, and all sort of light humor aside, the important point is mezigdomide as an oral therapy worked in triple-class refractory patients, and not only in those in whom all of the major drug classes had failed them, but also in patients who had BCMA exposure and indeed their BCMA therapies run out of steam. So we were dealing with an oral agent where you do it 3 weeks on, 1 week off. The response rate overall was around 41%, and everyone would say, well, to 60 or 70%. Well, it does actually, because you can just take it as a pill, and above all, we're going to be combining it with other agents in the future. And then when we looked at particular subgroups like the BCMA-exposed patients, in other words, those patients in whom either belantamab had failed them, or bispecific had failed them, or actually even a CAR-T had failed them, we were very impressed. The response rate was 50%, Jennifer. So that was really interesting to us, because almost suggests that a reset has happened, and in the context of and I want to be careful about that because I'm not sure we know that, but because of the immune activation hallmark of this drug, it does seem a reasonable hypothesis that in the BCMA-exposed patients, mezigdomide may be even more powerful. Now, iberdomide has also done great things. It's not as potent as mezigdomide, but it's moving earlier, and I want to especially acknowledge my colleague Sagalonial, who's led that work, and all of the team involved. And with iberdomide, what's happened is we've gone earlier, and it's got none of the side effect profile that can be sometimes a bit of a problem with lenalidomide, which is exciting. And what's really interesting as well in terms of iberdomide and mezigdomide is that they don't actually they don't seem to have the same risk of second cancers, which is extremely important. So that I think is very relevant. That becomes particularly relevant for iberdomide being used early and as a maintenance drug in the future, we hope.
Yeah, absolutely. And maybe one more, and then we'll go to questions, if that's okay. What about, I know Takeda is working on a drug called TAC573. Do you want to share what that is and how it works?
Yes, TAC573 is an immunoconjugate, so it works by targeting CD38 and then bringing an interferon warhead along with it. So a little bit like belantamab mafodotin, but a different target, CD38. So it's a it's a good drug and it's showing promise. And I think this targeted sort of approach, Jen, is really important because there are studies ongoing in that. And then, as you and I have talked about before, there are, other targeted approaches which deliver, cytotoxic warheads to where they're supposed to go. Mm-hmm. And of course, we also have, we have in Europe. It's not, unfortunately, hopefully back on the research table soon in the U.S. We have, of course, melflufen, which is the targeted way of delivering a cytotoxic warhead. So there are lots of excitement there. But pivoting back to TAC573, it's highly immunogenic. It appears to work very well with gamma interferon. And in the same way as belantamab mafodotin may be immunogenic, I think belantamab is, TAC573 is very promising and well tolerated as well in my experience. And also, for that matter, melflufen is also immunogenic. We've learned that, and I think that's really important to know. So these approaches all have an immune basis as part of what they may do, but some to varying degrees may be less dependent on the immune repertoire of each patient, which I think, again, pivots back to this key point, Jen, that we're able to offer different options to different patients in different settings.
Right, and at different stages. And I know, like for the melflufen example, it worked quite well in patients who had not had a prior transplant or a recent transplant. So,
yes,
and for patients who aren't going there, maybe that's not.
Well, Jen, I suppose the most important thing for melflufen, to be honest with you, is the elderly, right? The frail and elderly. I mean, it's a very well tolerated drug, and I think there's no mucositis. Patients don't lose their hair. It's a once-a-month infusion, and I don't know if you saw this, but in Europe, it was approved for peripheral infusion now. So it's got, even because previously we had to give it by central line. So I totally agree with you. I think in an older, frailer patient, it has promise, as shown in the clinical trials that led to its full approval in Europe. But I think, obviously, we're very hopeful we can get it back on the research agenda here in the U.S. and then move forward. But, I really wanted to emphasize to your point, though, Jen, that with drugs like TAC573 and belantamab, where they are so useful, in my opinion, is that they can be sandwiched into the sequence of treatments that we have. Mm-hmm. And we've got to think about how we use our treatments strategically to improve outcomes. So you can think about, if you've done a CAR T and the patient, unfortunately, the CAR T runs out of steam, how do you go straight to a bispecific, or can you give them a little bit of a break, give a patient a break to let their T cells reboot? What can you plop into that therapeutic sequence that will be effective, but at the same time not exhaust T cells? And so one example might be TAC573, or actually, for that matter, depending on your healthcare jurisdiction, melflufen or belantamab mafodotin. So you see how these things could be reasonably used.
Yes, absolutely, and I think that's where it's such a blessing to have all of this in myeloma. It's incredible what's happening, and thank you for doing the whole description, because it is so much to cover in 1 hour. I'm just going to open it up for a few minutes of caller questions. So if you have a question for Dr. Richardson, please press 1 on your keypad, and we have a first caller question will be 6251. Go ahead. With your question. Hello? Go ahead with your question.
Hello? Hello, Jen?
Oh, I'm here, but I thought we had I think they might have hit the button on accident. Okay. Is what I'd just like to ask is, as we move forward, what are you the most hopeful about in multiple myeloma and in the next, I would say, 2 years?
Well, I think, obviously, what I think we're seeing is that across the spectrum of disease, we're seeing an increasing proportion of patients enjoy excellent disease control for prolonged periods of time. What I'm very hopeful for is that the promise of the bispecifics and the CAR T treatments will lead to a subgroup of patients being functionally cured, essentially, by these approaches. I also think, by the same token, and it's my strong research focus, that we can complement complement the success of the CAR T and bispecific space by rationally building powerful oral agents, mezigdomide being one example, that can enhance our therapeutic armamentarium to further extend disease control in a way in which quality of life, above all, is also conserved or enhanced. And then at the same time recognize that all of the various drugs that we have, we need. So, again, small molecules, to my mind, matter. We touched on selinexor earlier. That may be I should have mentioned that actually as a potential sandwich for some of the strategies we just talked about without T-cell exhausting. So that's very real. I think, Jen, other aspects of this are the ability to have all the available agents to rationally combine and use in select settings, because, frailer older patients, to my mind, are an area we need to focus on, actually, because I think whilst these exciting new advances really have promise for the majority of patients, there are a significant number of patients who remain perhaps less able to safely receive some of these more intensive treatments, and to be able to offer those patients very safe, reasonable, non-toxic approaches that are convenient, to my mind, is very exciting. So I'm really excited by myeloma. It's been a privilege to be part of the research community for the last 25 years or more, and I think at the same time I'm so excited to see such incredible, exciting younger investigators really leading the charge with, cellular therapy, with bispecifics and so on, and really making a difference for our patients. And it's also wonderful, frankly, Jen, to have such great advocacy from organizations such as yours and your own leadership of your team as a patient who really knows what all this means, providing such service to our patients, because that to me is incredibly important too.
Oh, well, thank you so much. And for 1 minute, let's just go back to selinexor, because you mentioned it for deletion 17 patients earlier in the show. But are there other instances or sequencing that you typically use that?
Well, I think the important point to realize is that, again, some of the data emerging. For example, at this year's IMS, which is next week in Greece, we'll be talking about a subset from the Boston trial who were bortezomib naive, and they received bortezomib and selinexor in first relapse and enjoyed an amazingly long PFI. I mean, it's approaching 30 months, which I think is amazing. So. These weren't necessarily 17p selected patients. So, again, if you said 30 months in CAR T, they'd all say, oh, yeah, well, that's what we see anyway, isn't it? But selinexor and bortezomib in first relapse, 28 months median PFI, it's not bad, So
yes,
I think
totally.
there are ways to think. And I think, so selinexor, because I think what's most important about Seli is we've learned to give it in a way that's much more tolerable. Selinexor is a tough drug orally if it's given according to the label of twice a week and just with steroids. If you combine it with other drugs and give it once a week and go low and slow, as I like to say, it really, in my experience, can be very beneficial. In fact, I have a wonderful patient of mine with 17p disease, and he relapsed, unfortunately, after a platform of carfilzomib, isatuximab, lenalidomide, and dexamethasone. So he got excellent therapy up front. He got stem cell harvested. He wanted to keep transplant in reserve. He's got a young family, and he's a very busy executive, so he has a lot on his plate. And we got a wonderful result using selinexor, bortezomib, POM, and dex for him. Because he's 17b deleted, I should emphasize, and so that is relevant. But we were able to on-ramp him successfully to a CAR T, which we're hoping to pursue next month for him. So give you a real example. Lovely guy, lovely family, and oh my goodness, 5 years in, with bad high-risk disease at the beginning, hasn't needed a transplant, doesn't want it because he wants to be, fully functional and not have time away from his family. And we're very hopeful that the cilta-cel platform he'll be getting next month will take him to the next step.
Well, that's a perfect example for all patients. Well, all of us will have to consider a lot of these therapies as we have our life with myeloma, and hopefully one of these combinations and sequences will ultimately relate to a cure. So thank you so much, Dr. Richardson, for everything you are doing in development in the CAR-T, in the clinical trials on all platforms. It's just unbelievable, and I really look forward to hearing from you and many others at IMS next week.
My pleasure, Jen, and thank you for all you do, and above all, a huge thank you to our audience for taking time out on their Friday afternoon, and just wish everyone a lovely weekend. Thanks so much, Jen.
Yes, thank you so much, and thank you to our listeners for listening to the HealthTree podcast for multiple myeloma. Join us next time to learn more about what's happening in myeloma research and what it means for you.
Hello, it is Ryan, and we could all use an extra bright spot in our day, couldn't we? Just to make up for things like sitting in traffic, doing the dishes, counting your steps, you know, all the mundane stuff. That is why I'm such a big fan of Chumba Casino. Chumba Casino has all your favorite social casino-style games that you can play for free anytime, anywhere with daily bonuses. That should brighten your day a little. Actually, a lot. So sign up now at ChumbaCasino.com. That's ChumbaCasino.com.
No purchase necessary. VGW Group. Void where prohibited by law. See terms and conditions. 18+.